right? When we're talking about polypeptide, a protein, they're no longer amino acids, they're remnants. Okay, so that was hard to understand and confusing. Sorry. I think that is important stuff, and I want you to know it, but it's not going to be on your exam because it's challenging material. Let's talk about complex 2 now. Succinate Q reductase. Remember, this thing is huge. It also has many, many subunits, but it also has the enzyme succinate dehydrogenase, which is part of the citric acid cycle. It oxidizes succinate to fumarate, generating FADH2. And so what's going to happen is in complex 2, that FADH2 is going to donate electrons to, you guessed it, coenzyme Q. So it's working almost the same way as complex 1. The major difference is when you look at these tables, oh boy, they don't have FADH2. You guys. Remember, they're making life hard on you guys. This table's reversed. Let's go to 18.1. It's worth it because that's what you guys are going to be using. I'll just have to find my way back home again. Just notice that NADH has a more negative reduction potential than does FAD by a substantial amount. That means that the oxidation of FADH2 will result in less free energy available to drive conformational changes. So while complex 2 can catalyze the oxidation of FADH2 sufficiently that the transfer of those electrons to coenzyme Q is spontaneous, generating QH2, there's not enough additional free energy available to pump protons through the series of conformational changes that we just learned about in complex 1. So, okay, back to it. And so that explains why when you talk about FADH2, ultimately its oxidation will produce less ATP at the end of the day when we're finished talking about ATP synthesis. This slide is summarizing what I'm saying. You have a relatively small change in free energy, minus 2.9 kilojoules per mole, and that's just not enough to drive unfavorable conformational changes that result in the proton. Okay, so here's complex 3. It's called Q-cytochrome C oxidoreductase. It's another dehydrogenase. Its job is to catalyze the transfer of electrons from QH2 to cytochrome C, which we learned on Tuesday is a soluble carrier of electrons that exists in the intermembrane space. Now, one of the things that's interesting about complex 3 is we're going to be dealing with a carrier of 2 electrons, coenzyme Q, and a carrier of electrons that can only, and a protein that can only carry 1 electron, cytochrome C. So there has to be some mechanism to deal with this. And that's what's referred to as the Q cycle. That's what we're going to cover now. Here's the reaction. QH2 is going to end up reacting with 2 cytochrome C oxidases. And it's going to form oxidized Q, 2 reduced cytochrome Cs, and we're going to end up with 4 protons in the inner membrane region. And 2 protons are getting soaked up from the mitochondrial matrix. So let's see why. Where are these 4 protons coming from? And why are 2 protons being sucked up from the mitochondrial matrix? You guys can ignore this slide. It's just showing you the structure complex 3. So here's what I think is important to understand, is how does this Q cycle work? There's actually two different halves of the Q cycle. There's the first half and the second half. So in the first half, what's going to happen is a Ch18.2 from the Q pool. Remember, there's this thing called the Q pool, which is it's a mixture of oxidized Q and reduced Ch2 that simultaneously exist solubilized in the hydrophobic mitochondrial intermembrane. And so when Ch2 is present, it will then bind to complex 3. Complex 3 will also bind to oxidized cytochrome C. And remember, we've got this conundrum of having 2 electrons to deal with, but we have a carrier that can only receive 1 electron at a time. So what happens is the 2 electrons go on different paths from Ch2. 1 electron will go to cytochrome C, and another electron will go to oxidized coenzyme Q that binds in a site that is distinct from the Ch2 binding site. So we're going to end up generating a semiquinone radical here. Radical. And that's stabilized there. It's not going to go anywhere. So now we can already see how 2 of those 4 protons that end up in the intermembrane space got there. They came from Ch2. When Ch2 is oxidized, those protons are somehow channeled to the intermembrane region, right? So after the first half of the Q cycle, we've generated 1 reduced cytochrome C. We've fully oxidized 1 Ch2, and we've generated this Q radical. So that's the first half. So in the second half, what's going to happen is this Q leaves, joins the Q pool, a fresh Ch2 will bind, a fresh oxidized cytochrome C will bind, and now this Ch2 gets oxidized. It transfers 1 electron to cytochrome C. That's why we have 2 reduced cytochrome C in our balanced equation. The protons from that Ch2 will again go to the intermembrane space. That's where the 4 protons in the intermembrane space come from. And the other electron will go to the Q radical, filling up its electrons to be reduced. But remember, a reduced coenzyme Q also needs protons. And those 2 protons are going to come from the mitochondrial matrix. And that's why we're consuming 2 protons from the matrix. So this is a very elegant solution to the problem of how do we transfer 2 electrons to a 1 electron carrier. And as a consequence of this, so whenever coenzyme Q is getting reduced, either by FADH2 or NADH, it ultimately ends up in the generation, helping towards the generation of our proton gradient. Because the protons of QH2 will always end up in the intermembrane space. Not only that though, the Q cycle will also result in the depletion of 2 protons from the mitochondrial matrix to help form QH2 in the second half of the Q cycle. So I realize it's quite complicated, so I'm relying on you. I have to go fast today. If you're confused, please go to the book and make sure you get this. It's important. Last but not least, we have complex 4, cytochrome C oxidase. So as the name implies, what's going to happen is cytochrome C, the reduced form, will interact with complex 4. The electrons will travel down this really complex path to an oxygen binding site. And that's ultimately where oxygen is now accepting those electrons, generating water. All right, so did you guys know that you are powerful water generation plants? You're generating water right now through this process. And look at that delta G naught prime. Holy smokes. That's crazy. So it's not surprising that this complex can also act as a proton pump. And so it will ultimately be moving 8 protons out of the mitochondrial matrix. 4 of those protons are necessary to help reduce O2 to make water. We're going to generate 2 H2O. So that's where 4 protons are necessary. And then the other 4 are going to get pumped across the inner membrane into the intermembrane region. And you do not need to know this. This slide is just summarizing what I said. When cytochrome C in the reduced form is transporting electrons through this enzyme to oxygen, 4 protons are needed to form the 2 water molecules, and then 4 protons are able to be pumped against the proton gradient into the intermembrane region. Why? Because the proton pumping is coupled to the extremely energetically favorable oxidation reaction of cytochrome C and the reduction of oxygen. Okay. So here is the summary. And again, it's to a large extent, this is brute force memorization. The only thing that's not brute force memorization was my treatise of the pumping by NADH dehydrogenase, and I told you that as much as I want everyone in the class to be able to run around the world and tell everyone how that works, I think that it borders on the level of graduate school. And so if you got it, then it's amazing. If you're on the cusp of getting it and you want someone to talk to you about it, I'm here. Yeah. If I'm disturbed over my break by students in this class, that would not be the worst thing ever. Okay? So we have 4 complexes. Complex 1 is going to catalyze the oxidation of NADH. Complex 2 is going to catalyze the oxidation of FADH2. The complex 1 reaction releases more free energy. So in addition to generating QH2 for the Q pool, there's sufficient free energy to catalyze a series of conformational changes that make it favorable to pump protons against the proton gradient. In the case of complex 2, that free energy is not available, so only QH2 is generated. But as QH2 is generated and the ratio of QH2 to oxidized Q is high, that QH2 will bind to complex 3, where electrons are ultimately transferred to cytochrome C, resulting in the transport of protons from QH2 to the intermembrane region, helping to generate our proton gradient. I should remind you, by the way, that this thing about FADH2 is not that big a deal. Remember, when you're starting from glucose, we're going to get 8 NADH, I believe. Wait, 10 NADH, and 2 FADH2. So, you know, for the most part, we're dealing with NADH, and we're going to pump protons as a consequence. The QH2 that's generated will then help generate a proton gradient, and then the oxidation of cytochrome C, ultimately by molecular oxygen, releases a ton of free energy, and rather than that free energy just being released as heat, it's coupled to unfavorable conformational changes that result in the pumping of more protons, helping to generate this proton gradient. This is the kind of thing you're going to see on your exam. So I'm not going to waste my time with this. You guys are more than capable of handling this. Okay, so now we have our proton gradient. And remember, the mitochondrial matrix is going to be the inside of the mitochondria, And then it's separated by what we call the inner membrane space, the space between the outer membrane and the inner membrane. And that's where the proton concentration is high. And so, this stuff was figured out a long time ago by true genius level scientists. Think about it. Back in the day, they were like, oh man, we really need to explain how ATP is generated, right? That's an important problem, and I'm sure they were like, I might win a Nobel Prize one day, which would be sweet. Who would win one? They give you a ton of money. Sweet. I think I'd probably rather win an academy award. That's just me. All right, so what is this chemiosmotic hypothesis that was posed? Basically, they posed that the enzyme responsible for generating ATP synthase, is powered by this proton motive force. That somehow the free energy available in the proton gradient can be utilized to generate ATP. And when this was first posed, they were like, you're crazy. Right? So the scientists that believed in it had to actually prove this. Now, we don't have time today to go into the details of how this was shown. It's pretty cool though, I suggest you look it up. What we're going to do is just dive right into the mechanism of ATP synthase and how this machine is built, what are the components, and how do those components ultimately drive an unfavorable reaction through the release of the proton gradient. First thing first, you can see it's a multi-subunit enzyme. Part of the enzyme lives in the membrane. That's the inner membrane. So this is the inner membrane space. That's where the protons are. This is the mitochondrial matrix. So part of this thing is bound to the membrane. And then part of it is not bound to the membrane. This alpha-beta ring, it's a heterohexamer. It's going to have 3 active sites that are capable of stabilizing the transition state for ATP synthesis. And then these things are connected. They're connected by this gamma subunit and by this A, B2, and delta subunit. And the connection between these two things could be of no more greater importance. Insanely important. So they named the regions F0 and F1. The F0 component is in the membrane and the F1 is in the matrix. So let's first talk about F1. Consists of 5 different types of chains. Alpha, beta, gamma, delta, epsilon. And as I already told you, the alpha and beta are arranged in a hexameric ring. The beta subunits have the active sites. Between the alpha-beta subunits, please think of the alpha-beta ring as a donut with a hole in the middle of that donut. And in the middle of that is a stalk consisting of gamma and epsilon. Right? So we have I guess I'm just going to use donut analogies for the rest of the day. We have a donut, and then the gamma subunit can be thought of as a maple bar shoved into the center of the donut. And then I'm making a point here. I'm saying that the gamma protein has 3 different faces, sides. So just like I have my front and back, right? You can imagine also some object that had 3 distinct faces. I don't know, maybe one face is hydrophilic. Maybe one face is positively charged. Maybe the other face is hydrophobic. That's what they mean by that. So what happens is when the gamma stalk is inserted into the donut, each one of those faces will be facing a different active site. In other words, the hydrophobic face would be facing the active site. The positively charged one would be facing an active site that's distinct. And then the hydrophilic one is facing the last active site. So all three alpha subunits with their active sites will interact with this gamma stalk. However, the nature of those interactions will have different effects, as we will see shortly, on the active site.
So the alphas have three different faces themselves?
No, the gamma has the three different faces.
Oh, the
The
gamma.
alphas are identical.
That's what I thought.
Yeah.
Sorry.
Yep. And then F0 has what's called the C ring. So what we'll see is the C ring is to a large extent alpha helical, and it's a oligomeric. And so it's going to be arranged depending on where you live in the kingdom of life. Your mitochondria have anywhere between 10 to 14 subunits of this thing. And no matter what, they all close around each other to form a ring. That's going to be our different donut. We have our alpha beta ring in the F1 subunit. That'll be our chocolate donut. Whereas the C subunit ring, I don't know, will be our old fashioned. Never met a donut I didn't like. Okay, so now we're ready to talk about this reaction. So here are the substrates. This is the transition state. And this is our product. And they're just using R for the ribose and adenine. And so what's got to happen is the enzyme is going to have to, A, bind to substrates, B, stabilize the transition state, and C, let go of the product. And that's going to end up being the critical thing here. And that's what this slide is. These were experiments performed in the lab of Paul Boyer at UCLA, who was this super nice old dude when I was there, earning my PhD, this incredibly humble guy. And decades before I got there, he figured out a way of asking these kinds of questions. I told you guys earlier that this is very much like myosin. In the myosin, in chapter 9, we learned that in myosin, there was this curious thing where if you took radiolabeled water, O18 labeled water, you could use that as a way of understanding the equilibrium constant for ATP versus ADP plus orthophosphate on the enzyme. We already know what that equilibrium constant is in solution, right? It's whatever it is so that you get a delta G naught prime of minus 30 kilojoules per mole. But these clever experiments with labeling showed that if you incubate all of these different things in a test tube, labeled water, the enzyme, and ATP and ADP plus PI, you could figure out the equilibrium constant on the enzyme by just seeing how often that O18 gets incorporated into the ATP. And that's how they figured out that the equilibrium constant was not consistent with what we would expect based off of our measurements of the equilibria between ATP and ADP and orthophosphate in water. Instead, the equilibrium was about 1. And that was a game changer event for how we understand how these molecular machines work. That's why we say over and over again, when we teach you all, that the way enzymes function is by binding most tightly to the transition state. So the enzyme has evolved to bind most tightly to this, such that when these things are bound, all of them down to the enzyme, this is what you're going to see predominantly, because that's where the free energy is lowest, because of all those weak force interactions between the enzyme active site and the transition state. So then that led to a serious conundrum. When they did this experiment and they realized it's not, the free energy is not needed to stabilize the transition state, that's what led to the correct hypothesis that instead you have to pry the jaws open. That, in other words, what's happening is that in the absence of a chemical gradient, the enzyme has evolved to just bind in the transition state and clamp down. And it's not going anywhere, right? So to get the ATP off, something's going to have to create an unfavorable conformational change to go like that. It's like a bear trap. And it's like pulling the bear trap apart, and then now the ATP can diffuse away. And so that's what then, once that elegant series of experiments, biochemistry experiments was done, it was now, the ball was in the court of the structural biologists to try to figure out how is the movement of protons from the inner membrane space to the mitochondrial matrix coupled to an unfavorable conformational change, prying the active site open, enabling dissociation of substrates and products. And this led to what's called the binding change mechanism. In that model, the three, I made a mistake. It's the beta subunits that have catalytic activity, my bad. The three different beta subunits are going to exist in three different conformations depending on their interaction with the different three faces of the gamma stalk. Those three conformations are going to be called loose, tight, or open. The loose conformation is capable of binding to ADP and PI. The tight conformation stabilizes the transition state and will therefore catalyze the reaction. And the open conformation is when the bear trap has been pried all the way open, allowing ATP to dissociate. And so what actually then couples the free energy of protons moving through this machine on their way from the inner membrane to the mitochondrial matrix, it is going to be the driving of the rotation of this gamma subunit, where it basically just goes around like this, facing beta subunits. You know, so if Jacqueline is beta subunit 1, Chase is beta subunit 2, and then the table, sadly, is 3, right? The gamma stalk is going like this, this, and it keeps doing it over and over and over. And so ultimately then the question is, if that's true, then how does the movement of protons drive that motion of the gamma stalk? So this slide is just summarizing with an image what I said. So they're trying to show you that the gamma stalk has three different interfaces that face the beta subunits. The pointy one, the concave one, and the flat one. And so when the flat one interacts with this beta subunit, it's in the loose conformation. When the pointy surface of gamma points to this beta subunit, it's in the tight conformation. And then when the concave surface interacts with this beta subunit, that's in the open. And so what they're showing is through the rotation of gamma from here to here, and then again and again, what's going to happen is we're going to go from this active site, where it's tight, rotation of gamma I guess it's counterclockwise, right? So we ended up here, is now going to change the conformation of this active site from T to open, enabling ATP to leave. Similarly, this active site, which was loose, which enabled binding of ADP and orthophosphate, that is then going to become T when the gamma shaft rotates and the pointy end is facing this active site. And now we're going to have stabilization of the transition state. And so on and so forth. So when this thing is now pointing over here, this beta subunit will change its conformation to open, enabling the ATP to leave. So through the spinning of this gamma subunit, we're able to catalyze unfavorable conformational changes in the active sites of ATP synthase. And so now we just have to try to understand how does the flow of protons turn this thing in the first place. Yeah?
So wherever the gamma subunit is pointing is the one that's tight.
I wouldn't think of it that way. I would think of it as there are 3 different surfaces on the gamma subunit. And each one of those surfaces is facing an active site and has 3 different effects on the conformation.
Oh, I see that one. I'm trying to just look at it.
Okay. All right, so now let's address how does rotation occur. So now we're looking at F0, the F0 segment. We have our ring, our old-fashioned donut of C subunits. And that is going to interact with what they call the A subunit, shown in purple. And the A subunit is going to have 2 different channels, one facing the intermembrane space, and the other facing the mitochondrial matrix. And here's what's going to happen. Protons are going to enter into the half channel facing the intermembrane region. They shouldn't say cytoplasmic, they should say consistent. Intermembrane space. And the flow of protons down that channel will end up resulting in the interaction of this half channel with a proton binding site on the C ring. So each subunit of the C ring is going to have an aspartic acid residue that is capable of either being protonated or deprotonated. When it's facing this half channel, it gets protonated. When it's facing this half channel, it gets deprotonated. So protonation of that residue is energetically favorable here, but unfavorable here, which is why the proton will dissociate. And so what ends up happening is because it's energetically favorable for the proton to dissociate from the C ring, from here, what's going to happen is the C ring turns, right? Because every time it turns, effectively, a proton will now bind here to an empty C subunit and will dissociate here from a occupied subunit. And let me show you now why that's the case. So this slide is just showing you what a C subunit looks like. It's 2 alpha helices interacting, and here is the aspartic acid, where we now can say, in the language I taught you, that the pKa of that aspartic acid is going to be different in these 2 different environments. Here, where the environment is facing the intermembrane space, protonation is feasible because of that pKa and, of course, the concentration of protons in the intermembrane space. It's a combination of 2. Whereas in this environment, protonation is not feasible. There's going to be a difference in those pKa values. And how is that manifested? Through the control of the environment of the aspartic acid in those 2 different environments. So this is an image trying to show you how turning of the C ring will occur. We start here where a proton will now enter the half channel of the A subunit, bind to the C subunit facing that half channel. And there's one more important thing I need to tell you guys to understand this. The binding of that proton changes the chemical nature of the aspartic acid side chain. So what is that side chain? It's this. And when it gets protonated, it becomes this. So which one of these species is less favorable in terms of its interaction with a hydrophobic environment, which the membrane most certainly is? Which one would you choose on your exam if I said which species will be less favorable in terms of its interaction with a hydrophobic environment? You would say the COO minus, because negative charges that are unpaired are extremely unfavorable. In water, we're good to go, because the water molecules will just congregate around that negative charge, forming hydrogen bonds, stabilizing it. This, on the other hand, even though it's still somewhat polar, can interact with the hydrophobic environment of the membrane. So when a C subunit aspartic acid gets protonated, it will now enable the rotation of that C ring, because now that proton is going to move over and face the hydrophobic portion of the membrane. And that's not going to be unfavorable anymore, because the negative charge is now gone. And similarly, as the C ring rotates, the proton that exists on this C subunit is now facing the channel that is also facing towards the mitochondrial matrix. The proton falls off. Why? Because it no longer in that environment has great affinity for that proton. Here, it has affinity. Here, it doesn't. And so, because this is energetically favorable, the C ring is just going to rotate. And because the C ring is connected to this gamma shaft, the gamma shaft turns. So as long as the alpha beta subunits, let's look at a picture of this thing. The whole smash thing. Where are you? So as long as this alpha beta heterohexamer is held in place relative to the gamma shaft, then the turning of the gamma shaft is going to result in these conformation changes in the active sites of the beta subunits. So how is the alpha beta subunit held in place? Through the connection of this A subunit and this long B2 shaft and delta. Right? So these things basically are all connected, ensuring that the only things that are turning is the C ring and the gamma. So cool. All right. So now we're finally ready to start doing some calculations. What are we getting out of all this in terms of the ATP field? So I told you already that when you look at ATP synthetases across life, you're going to find anywhere between 10 to 14 C subunits per C ring. And we now know that the complete rotation of the C ring and the gamma shaft will ultimately result in the production of 3 ATP. Right? Because as we go around, each 120-degree turn will result in the opening of the jaws, allowing 1 ATP to dissociate. So 3 120-degree turns, or a full circle, will open 3 active sites, generating 3 ATP. So if it takes 1 proton to move each subunit, and if there are 10 subunits, we need 10 protons to fully turn this thing. So that means that what we're getting is for every 3 protons that travel through ATP synthase, we're going to generate an ATP. And that tells us now the efficiency of ATP synthesis versus generation of the proton gradient. And through calculations that, frankly, the book doesn't go into, they make some, based off of what I just told you, if you actually know how many protons are pumped across the membrane upon the oxidation of NADH or FADH2, you can get an idea, if you make a ton of assumptions, how many ATP are generated. And when you do that, you can see that each NADH is going to generate approximately 2.5 molecules of ATP. And each FADH2 is going to generate approximately 1.5. So now what we can do is we can go through the citric acid cycle, glycolysis, pyruvate dehydrogenase, the electron transport chain, and ultimately understand how many ATP we're getting from glucose. So please remember that in glycolysis, during stage 1, we actually invested 2 ATP to get things going. But then, I don't know why they're breaking this up now into 3 stages, 2 is fine. In the second stage, the payback stage, we're going to generate 4 ATP. So 2 invested, 4 generated, so we have 2 net so far. Then, if we calculate how many NADH we've generated, well, I've got 2 here, I've got 2 here, and I've got 3 from the citric acid cycle, that's 10. So 10 times 2.5 is 25. So now I'm at 25 plus 2, which is 27. And then we can ask how many FADH2 are generated? 2. And so those 2 FADH2 are going to generate 3 ATP. Now we're up to 30. So we're almost there. We have one more event of substrate level phosphorylation here in the citric acid cycle. And that's going to generate 2 more ATP or equivalents of ATP per glucose, resulting in 32 ATP through the complete oxidation of glucose, through glycolysis, the citric acid cycle, and oxidative phosphorylation. So hopefully you can see now how glycolysis is rather pathetic in terms of its ability to generate ATP. It's good enough to drive the lives of anaerobic bacteria, good for them, but I think that's being an anaerobic bacteria sounds really boring. I think we've got it way better. And so this has enabled complex life because you're generating literally orders of magnitude more ATP as a consequence. And so I give you this slide to go through my calculus if that was confusing for you. And then you guys are on your own for how all this is regulated. It's extremely logical. It's the same principles that we've been talking about. So I don't think any of you should have any trouble thinking about this. And then last but not least, so we officially close GoodNotes for ever and ever. And then I want to watch this sweet video. And I'm going to have to narrate. So this is showing you, I have terrible resolution, sorry. Let me see if I can fix that. Oh, there. Whew, that was easy. So that's the gamma stalk. It's turning, and this is the membrane. So there's our C ring, and there are our beta subunits for the active sites, and these are little substrates, ADP and orthophosphate, and ATP products. So I'm just going to show us this now in greater detail. So those are protons, the little ping pong balls. Now they're showing us in atomic resolution detail the conformational changes that are occurring between loose, open, and tight as that gamma stalk is turning. So here we have ADP, our friend. There's the two phosphates. And then now we're going to be looking at weak force interactions between ADP and the enzyme. And it's not surprising that there are many of them. There's a magnesium ion, that's part of the mechanism. You guys know about metal ion catalysis, so that should make immediate sense to you what that is doing there. And then now they're going to show us the binding of inorganic phosphate and how it changes during the transition state. And look at how the residues in the active site were adapting. They were coming in and binding to that transition state, greatly stabilizing it. And those conformational changes are occurring thanks to rotation of the gamma stalk. So now they're showing us again in faster speed the whole process of this gamma subunit turning, creating conformational changes in the beta subunit. Now they're going to show what's happening in the A subunit and the C ring. So there's protons leaving, but now they're going to go in and show us binding of those protons to the aspartic acid. So there's a lot of protons in the inner membrane space there, right? Tons of them. And since the pKa of that aspartate is favorable enough for binding, the protons will then go in, bind to the aspartic acid, travel all the way around till they're facing the half channel that is exposed to the mitochondrial matrix, and now the affinity is changed, where the protons don't bind well anymore to the aspartic acid. They dissociate off. And this is the mechanism for how turning of the C ring and therefore the gamma shaft occurs. This is a beautiful view now showing the conformational changes in the three different active sites as the gamma shaft is rotating. All of this is based off of atomic resolution crystallographic structures, which is amazing. And then something bad happened. Something got stuck. And I don't know if this video has. There's one more thing I wanted to show you guys. I don't know if they show you right now. There's somebody talking, saying a bunch of stuff. So let's see if I can show this or not. This should be better. Okay, so let me just make one quick point. We're almost done. So, in X-ray crystallography, you're not going to see turning. These are all snapshots of rigid protein structures. And so, you guys know how they were able to get those different structures. You know about transition state analogs and how you can then put those into or bind them to your protein, and then this will show the conformation of beta subunits when they are in the tight conformation. And then you can solve the structure in the absence, right, of these things, or in the presence of other things. But how do we really know this is turning? And so the critical experiment that was done that is like beyond awesome is they recombinantly generated a gamma shaft that was linked to a long arm made out of protein, and at the end of it, they put a fluorescent molecule called green fluorescent protein. So effectively, they generated a gamma shaft, just like normal, and it interacts with all the different components of ATP synthase, but then it has this extra thing on it, this long arm, and at the end of the arm is a fluorescent molecule. So yeah, go ahead and look at the ceiling, and hopefully I won't pass out when I do this. But what they did is they have microscopes that are powerful enough where They then apply these ATP synthases to a microscope slide, and then cleverly they add ATP. Now ATP synthase becomes an ATPase, right? Because in the absence of the proton gradient, it's going to do whatever is most energetically favorable. A hydrolysis of ATP, and they literally saw this in their microscope. I mean, the graduate student who did that, I'm like insanely jealous, because you really don't get to do that very often in life. So office hours, if you want to be there, I'll wear this. Questions, email me over the weekend. I'll be home drinking tea